Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the grand tapestry of scientific discovery, certain publications emerge as singular threads, altering the entire pattern of human inquiry. They are documents of such profound insight and consequence that the intellectual landscape is irrevocably reconfigured in their wake. The 2012 paper from the laboratories of Jennifer Doudna and Emmanuelle Charpentier, titled A Programmable Dual-RNA-Guided DNA Endonuclease in Adaptive Bacterial Immunity, is unequivocally such a work, published in the journal Science. This study represents a watershed moment, a pivot from the descriptive to the prescriptive in molecular biology. It transmuted a fascinating but esoteric aspect of prokaryotic defense into a tool of breathtaking power and simplicity. The paper stands as a masterclass in curiosity-driven research, where the meticulous deconstruction of a natural mechanism yielded the blueprint for its revolutionary reconstruction, providing humanity with a means to engage the genome with unprecedented precision. The genesis of this breakthrough lies in decades of patient observation, a slow accumulation of seemingly disconnected biological puzzles. The story begins in the early 1990s with the work of Francisco Mojica at the University of Alicante. While studying the genome of the archaeon Heliothrax mediterranei, Mojica noted peculiar, repeating sequences of deoxyribonucleic acid interspersed with unique spacer sequences. He christened them clustered regularly interspaced short palindromic repeats, or CRISPR. For over a decade, these structures remained a biological curiosity, an intriguing genomic signature of unknown function. The first major conceptual leap occurred in 2005, when several research groups, including Mojica's, independently hypothesized that these CRISPR arrays function as an adaptive immune system for bacteria and archaea. They observed that the spacer sequences often matched the deoxyribonucleic acid of invading viruses, or phages, suggesting that the spacers were a molecular memory of past infections. Subsequent work from the laboratory of Luciano Marafini at Rockefeller University provided critical evidence that the CRISPR system in certain bacteria targeted deoxyribonucleic acid, silencing the genetic material of invaders. The field rapidly began to characterize the machinery responsible, identifying a diverse family of CRISPR-associated, or Cas, proteins. Among the various types of CRISPR systems, the type whose system appeared particularly streamlined, a pivotal piece of the puzzle, was laid in 2011 by Charpentier's own group. While studying the pathogenic bacterium Streptococcus pyogenes, her team discovered a small, previously unknown RNA molecule they named transactivating CRISPR RNA, or trcrRNA. It demonstrated that this trcrRNA was essential for the maturation of the CRISPR RNA, or crRNA, the molecule that contains the viral spacer sequence. This discovery of a third critical component in the Type II system set the stage for a landmark collaboration, one that would seek to fully reconstitute this bacterial defense mechanism outside the cell. The 2012 paper is the breathtaking result of that collaboration between Doudna's structural biology expertise and Charpentier's microbiological and RNA-focused research. Their central objective was to test a powerful hypothesis that three components, the Cas9 protein, the CRISPR RNA, and the transactivating CRISPR RNA, were the sole necessary and sufficient elements for sequencer-specific cleavage of target deoxyribonucleic acid. To achieve this, they undertook a series of elegant in vitro reconstitution experiments, a hallmark of classic biochemistry. The researchers painstakingly purified each molecular actor with Cas9 endonuclease, the crRNA containing a specific spacer sequence, and the trcrRNA. They then combined these components in a reaction tube with a plasmid deoxyribonucleic acid molecule engineered to contain a target sequence complementary to the crRNA spacer. The results, visualized through the straightforward technique of rigorous gel electrophoresis, was spectacularly clear. The presence of all three components together resulted in a precise, double-stranded break in the target deoxyribonucleic acid. The omission of any single component, be it the Cas9 protein or either of the two RNA molecules, abolished this activity completely. This confirmed their core hypothesis with unambiguous clarity. The genius of the work, however, extended beyond this confirmation. The paper meticulously dissected the molecular interactions. It revealed that the trcRNA performs a dual role. It first base pairs with the crRNA, forming a distinctive 2-RNA structure. This duplex then acts as a scaffold guiding the Cas9 protein to its destination on the genome. The crRNA component, specifically its 20 nucleotide spacer region, serves as the ultimate targeting device, scanning the genome and binding to a complementary deoxyribonucleic acid sequence. They further defined a critical constraint of the system. The Cas9 protein would only cleave the target deoxyribonucleic acid if the target sequence was immediately followed by a specific, short sequence known as the protospacer adjacent motif, or PAM, for the Streptococcus pyogenes Cas9. This sequence is NGG, where N is any nucleotide and G is guanine. This P or M requirement acts as a vital self-assessment and self-recognition mechanism, preventing the CRISPR system from attacking the bacterium's own CRISPR locus, which lacks the P or M sequence. Through this series of experiments, Doudna and Charpentier moved from a general understanding of a bacterial immune pathway to a precise, mechanistic blueprint of a three-part molecular Having fully elucidated the natural system, the researchers then made an intellectual leap of profound consequence, an act of bioengineering that transformed the discovery into a technology. They recognized that the complex, Dual-RNA guiding structure was an elegant product of evolution but potentially cumbersome for laboratory use. They hypothesized that the two separate RNA molecules, the CRRNA and the TRCRRNA, could be synthetically fused into a single, contiguous molecule without sacrificing function. This insight was the crucible in which the modern tool of CRISPR, Cas9, was forged. They designed a chimeric RNA, which they termed a single-guide RNA or sgRNA. This engineered molecule contained the 20-nucleotide target recognition sequence from the CRRN at its 5' end, seamlessly linked via an artificial hairpin loop, a simple tetraloop, to the Cas9 binding sequence derived from the TRCRRNA. This was a stroke of sublime simplification. The experimental test was direct and conclusive. They programmed the Cas9 protein with this new single-guide RNA and demonstrated that this simplified two-component system, composed of protein and one engineered RNA, directed cleavage of target deoxyribonucleic acid with an efficiency equivalent to the natural three-component system. This act of molecular fusion was transformative. It meant that to retarget the entire enzymatic machinery to a new deoxyribonucleic acid location, a researcher needed to synthesize a new, short single-guide RNA molecule. The protein component, Cas9, remained constant. The paper's title itself encapsulates this achievement: a programmable Dual-RNA-Guided DNA endonuclease. By the paper's conclusion, they had demonstrated its reduction to a single-guide RNA system, rendering the programmability astonishingly simple. The obscure defense mechanism of a bacterium had been successfully repurposed, streamlined, and unveiled as a universal deoxyribonucleic acid cleaving tool whose targeting specificity could be dictated by the straightforward principles of Watson-Crick base pairing. It was a discovery that carried the unmistakable resonance of a technological revolution. The legacy of the 2012 publication was immediate, immense, and continues to expand. While the paper itself was confined to in vitro experiments, its closing sentences clearly articulated the potential for genome editing in living organisms. This created an intellectual starting pistol for laboratories around the world. The clear, mechanistic instructions provided in the paper served as a direct roadmap for applying the system in more complex biological contexts. Within months, the promise was gloriously realized. In January of 2013, two papers published nearly simultaneously, one from the laboratory of Feng Zhang at the Broad Institute of MIT and Harvard and another from the laboratory of George Church at Harvard Medical School, demonstrated that the CRISPR-Cas9 system could be successfully deployed to edit the genomes of mammalian cells, including human cell lines. These studies, building directly upon the foundational biochemistry established by Doudna and Charpentier, confirmed that the simplified single-guide RNA system functioned with remarkable efficacy in the complex environment of the eukaryotic nucleus. This opened the floodgates. The simplicity and robustness of the CRISPR-Cas9 tool led to its rapid adoption in thousands of laboratories globally, democratizing the field of genome engineering. The technology has since evolved far beyond its initial function of creating double-strand breaks. Researchers have engineered nuclease versions of Cas9, or dCas9, which combined to a target deoxyribonucleic acid sequence without cutting it. By fusing these dCas9 proteins to transcriptional activators or repressors, scientists created tools for turning specific genes on CRISPR activation or off CRISPR interference at will. Further innovations, most notably from the laboratory of David Lilou, also at the Broad Institute, have led to the development of base editors and prime editors. These are sophisticated molecular machines that use a Cas9 targeting chassis to perform precise chemical surgery on individual deoxyribonucleic acid letters, converting one base to another without inducing a full double-strand break. The reception of the 2012 paper was consequently one of universal acclaim within the scientific community, but its profound implications also sparked intense ethical debates and a fierce, protracted patent dispute over the intellectual property of the technology. This very contest for ownership stands as a powerful testament to the universally recognized value of the discovery they had unveiled. In conclusion, the 2012 paper by Doudna and Charpentier occupies a hallowed place in the annals of biological science. It is a work that perfectly marries the spirit of pure, curiosity-driven inquiry with the vision of world-changing application. By meticulously deconstructing the components of a bacterial immune system, they revealed a molecular logic of stunning elegance. They then went a step further, applying a brilliant stroke of engineering insight to simplify the natural system, converting it from a biological curiosity into a universally programmable tool. This publication provided the essential mechanistic understanding and the key technological innovations single-guide RNA that catalyzed the genome editing revolution. It laid the intellectual foundation upon which an entire field has been constructed, a field that is already transforming medicine, agriculture, and our fundamental ability to understand and manipulate the code of life. The paper is more than a report of experimental findings, it is a declaration of possibility, a testament to the power of fundamental science to furnish humanity with tools that were once the exclusive domain of science fiction. Its legacy is written not merely in citations and accolades, including the 2020 Nobel Prize in Chemistry, but in the countless laboratories and clinics where the principles it first articulated are now being used to chart a new future for biological science and human health.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.